Okay. So first of all, thank you everyone for joining us today, and also for the help you have provided with us, or you're going to provide for this project. Um, I have to say, first of all, that all these slides I'm going to use, or most of the slides I'm going to use, have been generated by Rank over the last months. So I'm really thankful for that. Otherwise, it would have been much more complicated. So also, I would like to acknowledge all the people that, let's say, contributed to this effort in the past. There are a lot of names that are actually missing from this slide, I fear, but these summarize many of the contributions that were given, and that actually allowed us to be where we are now. So I have a list here of genomes that people, some people that are in the call, some others that may not join but will be able to listen to the recordings, have been assigned already. But as I said, there will be some more genomes coming in, and so for the people that have not like a genome assigned but would like to have one, we will have some more. So as you can see in this list, there are all fishes, with the exception of the lesser castle, which is a trio for which we are, the VGL is currently generating the PacBio data. One thing here, so most of these genomes are small, so they should be quite straightforward to assemble. We tend to have like issues when the genome size goes beyond like 2 to 3 gigs, but in this case it's like 1.7 gigs maximum. So why? Just a brief introduction to as to why we are. We have set up all this effort. Is why do we need and why do we want to have these high quality reference genomes? Of course, this will inform phylogenomic studies, or evolutionary studies that are carried out either by us as a consortium, or by other groups. Same goes for comparative genomics. Also, there are a few groups that are focused on the study of specialized traits that may also be relevant for biomedicine. So having high quality reference genome assemblies will definitely help in that. And also for conservation genomics, in particular, one striking example that we very recently assembled. The vaquita population is now limited to less than 20 individuals all over the world, and that's because of fishing, overfishing, that is also impacting the vaquita population. And what we found that was very interesting, and I think it's one of the reasons why this gene is that apparently it doesn't hold true that there is a inbreeding depression in vaquitas. So even with very few individuals, you may actually be able to recover the population. So when we talk about, when we say we want to assemble high quality genomes, we actually are targeting for specific metrics of the final assembly. So our sequence level goals is 1 megabase and 50 context size. For those who are not familiar with the concept of N50, is essentially a metric that somehow measures the average over a genome assembly size. So assuming that your genome assembly is, in this particular case, 400 megs, half of your assembly size, that's why we measure the N50 specifically. So 50% of the assembly size would be 200 megabase pair. And then if you order context, so the sequences that you have assembled out of your genome by size, and you start summing them up until you reach half of your assembly size, that's your N50. And that's a very popular metric, and we're going to measure it over and over again. Beware that it's not necessarily accurate sometimes, or it can be misleading in what's going on. But on average is giving you some hints on the quality of your assembly. So we are targeting at least 1 meg, and that's now with the pipeline we have. So now it's fairly easy to achieve in most cases, and should be so for most of your genomes. But if you don't, we can, see what's going on. Also, we're targeting QE40. Sorry, we're targeting QE40. Which means that there should be no more than one base pair error in the final assembly out of 10,000 base pairs. Also aiming at representing both haplotypes. This is part of the pipeline, so assuming that all our genomes that are vertebrates are diploids, we're trying to have a primary assembly representing a sort of complete assembly, and also some alternate haplotics, which represent wherever the genome is actually variable between the maternal and paternal haplotype. Then we have, as you see here, higher order goals that are potentially helpful for like synteny analysis, for reconstructing ancestral karyotypes and so forth. And for that, we are aiming for 10 megs scaffold N50. And also for at least 90% of the sequence to be assigned to chromosomes, identified chromosomes. So here, this slide summarizes how we are working at the moment. So there is data generation that is carried out mostly here at the Rockefeller, at the Sanger Institute in the UK, and at the Max Planck Institute in Dresden. There has been also some contribution from BGI, and if, let's say, some group generates data outside these sequencing centers, we also can use those data and generate assemblies. Most of these fishes that we are talking about at the moment have been generated, the data has been generated at the Max Planck. Then we move the raw data on GenoMark, as you can see here, and then we use, for most of the time, we use DNA Nexus to actually generate the assembly. DNA Nexus is a platform that is sort of layer on top of the Amazon cloud. So it's essentially using the Amazon compute resources or storage to store and compute on our data. And we established the entire pipeline there. And it's very helpful because now it has this graphic interface, so you don't have to like code or use command line for most of these steps, or even all the steps that we are carrying out during the pipeline. There is a very active assembly working group that is having like a Friday call every week, and this assembly working group is trying to design, assemble, collect, and organize all the data. Just a few words. So the data that we're uploading on Amazon Cloud within this GenoMark is public, so available for everyone to download. And also all the outputs that we generate, also, the final assemblies, but also the intermediate files that are generated are saved and stored on this GenoMark, and again, publicly available. Here's how the GenoMark currently looks like. You can look it up if you go to this GitHub website. So for each species, you have some information, let's say how much data was generated at the moment, if there is already an assembly or not. Within each individual species page, you can also see how many, how much raw data was generated for each technology that is involved in the pipeline and the approximate coverage. Also, whenever there is an assembly, you will notice this extra columns here, extra tables, where you have also the NG50, which is like somehow related to the N50 for those who don't know, but the NG50 is the N50 where instead of using half the genome assembly size, you use half the expected genome size. And this may sometimes be different if, let's say, part of your assembly is missing. So part of your genome sequence is missing from the assembly, especially when the repetitive content is high. And then we also have the mito sequence whenever available. You can also access the GenoMark if you're familiar with command line interface, using a command line interface from Amazon. And here at this webpage, you will find how to do that. So the pipeline, you probably have already seen this one. So this is the 1.5 pipeline. We have now moved to the 1.6. Some people here may have already used this 1.5 pipeline, and there has been some few changes which are summarized here. Essentially, one of the changes that was introduced is these porch tube steps, and also we changed something here when we combine the alternate and the primary scaffold, the alternate contigs and the primary scaffolds. But overall, I mean, it's pretty consistent. So I will now go through all the steps of the pipeline so that you can start familiarize with that. Also, sorry, this is the structure of the data when you go to the Amazon Cloud bucket, where we store all the information. So for instance, this is the Hummingbird example from the Hummingbird. As you can see, you will have one folder with all the genomic data like PacBio, 10x, Bionano, Hi-C, potentially also from different companies. And then whenever you will have the assembly available, you will also have a folder like assembly VGP standard with the final assembly that was, let's say, also went through the curation steps, but also all the intermediate files that were generated throughout the assembly process, so that we can go back and see what happened, let's say, at some point. We also have a trio binning pipeline, which is a different approach. I mean, it's an approach based on having also parental information, so the information from the maternal and paternal haplotypes, normally Illumina short reads. And what is interesting here is that it allows you to clearly separate within the deployed assembly. It really allows you to separate out the contribution from the mother and the father to the child haplotypes. So this is the pipeline for the trio, which, as you can see, is fairly similar to the non-trio pipeline, but has some differences, and also in terms of how the files are organized on AWS. So this will be relevant only for the Esther Castle at the moment, but there will be some trios coming in as well. So if someone wants to try that too, it's possible. The pipeline is available for command line users on this GitHub web page. This has been developed by mostly by Erang, and everything is there. So there is all the code. If you're working on this LARM, that's pretty straightforward to use, assuming you're familiar with that. But as I said, most of the people will now use the pipeline on DNA Nexus, which has this command line, this web interface, which is much more, practical. So you can drag and drop files, you can run everything from there. I know some people already got familiar with that, but I'm trying to make sure that everybody who is listening or will be listening to this video in the future will be able to start from scratch without any prior knowledge. So one thing which is important, so here, this web page here is a typical project on DNA Nexus, and in particular, this is the VGP tools. So it's like a project where we have all our workflows, or all our let's say all the code that is associated with our pipeline. And in particular, there are 7 workflows that we use to run the entire pipeline. As you can see here, now I'm going to go through all of them. So the first step, as you can see, the first one here is this Falcon and Zip workflow. Falcon is essentially the first step of the pipeline that is using only the PacBio data and aims at generate the primary and alternate haplotypes. So that is, as I said, the primary is a sort of the entire representation of the genome, while the alternate only represent wherever the genome is different between the maternal and paternal haplotypes. Why do we use long reads here? That's fairly simple. Long reads have a higher error rates, but on the other hand, being longer whenever you have to assemble a complex region of the genome that have, let's say, a high repetitive content, they will allow you to breach every of these repetitive elements and make your essentially, if you compare it to assembling a puzzle, it will be a puzzle with less pieces. So just to a very quick introduction to long reads. Long reads are single molecule technology as compared to any NGS technology, which has some level of amplification of the DNA usually. As I said, they provide long reads in terms of 15 to 100 1,000 base pair long, 15 kb to 100 kb long reads. On the other hand, to have a lower throughput compared to NGS, but this is also changing. So, as I said, so the first step, let me just go back here. So the first step, primary contigs and alternate contig haplotypes. That's what we're aiming for. And the way we do that, we are using the software from PacBio, so the same company providing the sequencing data. And this Falcon, and particularly Falcon Unzip, is the tool that we're using. So I'm not going to go through specifically into the details of our Falcon runs, but the idea is that you essentially select the longest reads that you have. They are acting as seed reads, and then you are aligning back all the shorter reads to those seed reads. You're using them for polishing, and then for polishing the long reads and all the reads, and then you're generating the layout of your contigs. Then the unzip part of this software, what it's trying to do is trying to say, okay, well, now we have contigs, but if we map back all our raw data, we notice that whenever the paternal and maternal haplotypes diverge, there will be a region where, of course, are not supporting just the assembly, but are also supporting some alternative sequence at that region, and is essentially trying to generate these haplotics at the regions where there is divergence. And that's why you end up having primary contigs and alternate haplotics. So here, the blue line represents your primary extended sequence, and the haplotics are represented by the red lines. Please notice here that this doesn't mean that the blue one is the paternal and the red one is the maternal, but they are actually mixed. But what you should be confident in is that the red line is a haplotic, so it's either paternal or maternal, that there is a mixture of, let's say, SNPs or structural variants within that particular haplotic. Then for running Falcon, also for these fishes that you have now, I mean, as I said, they have been generated at Max Planck, and they also generated already the primary and alternate contigs. So for most of these, you will not need to run this specific stack because it has been already run locally by Martin. But nevertheless, it's good to know. And for some other cases, you will have also to generate the contigs. So. And the only thing, I mean, the most relevant parameters that you will need to provide for this particular step is just the genome assembly size. And the reason for that is that when you're targeting the seed reads, you want to make sure that the seed reads that you have available are covering enough of the genome. And that's why you want to know the approximate genome size. And this is one, so this table here, you may have seen it already. If you haven't seen, it's a Google sheet where we summarize all the information that we have on the assembly that is ongoing, on any assembly that is ongoing. And as you can see, there is one column where someone extracted already from the literature the genome size that is being like predicted or estimated in some way. But also we have this column here where the genome size has been predicted based on the raw data. So there are means allowing you to predict the genome size based on the count of k-mers in your raw data. Sometimes it can be really accurate, other times maybe not. There is actually, so with DNA Nexus, there is one pipeline that is available to generate one estimate of the genome size, which is this one. So Jellyfish and GenomeScope It's one of the applets that are available through the Nexus, and as I said, it's just a web interface. So you just need to provide the raw data by selecting them from a drop-down menu. But then, so through this applet, you will be able to estimate with some accuracy the genome size. The problem of this pipeline here is that the one that is specifically using Jellyfish, whenever the genome size, whenever the genome is highly repetitive, it may actually underestimate the genome size, which therefore will lead to a wrong calculation of the required coverage for seed reads. So, okay. This slide is just to tell you that repetitive elements are the major driver for let's say the contiguity of your genome assembly. So here you see that essentially the white bars here, the more repetitive your genome is, the less likely you will be able to assemble it into a contiguous sequence. Okay. I'm sorry, I don't know if you see that, but I'm trying to turn it off. So, yeah, so that was the repetitive content. As you can see, the first step after you generated the contigs is that of purge dupes. So the purge dupes is something that I would say is most of the pipelines that are like out there. So what it's trying, actually these purge dupe steps is trying to target the problem of retained haplotypes, which essentially means that some of your alternate haplotypes have been, let's say the software is not able to identify them as being alternate, and it's actually retaining them into the primary. So imagine this is your primary sequence, and this is what was supposed to be the alternate. But at some point, because of, let's say, too high divergence between the maternal and paternal haplotype at that particular region, you end up retaining one of the alternates haplotype into the primary. And this leads to a sort of artificial duplication in your haploid. I mean, we're always targeting haploid assemblies, so it's going to have an additional copy of some region of the genome. And this was observed. That's something that you can see here. This was observed in some cases, and that's why initially there was this purge haplotics, but now we are using this purge dupes that seems to be more effective at removing these sequences, and is also taking care of some additional problems that the assemblies at this stage may have. Yeah, so that's pretty much it. So then you will, as soon as you have, let's say, purged all these alternate haplotypes from the primary, you will end up having your purged primary, and then you will also have this alternate which potentially will also contain what was purged from the primary. We are going to only scaffold the primary, so we're going to take our primary and we're going to go through all the scaffolding. Scaffolding, which means that you actually put together contigs, smaller contigs into larger scaffolds that are separated by runs of ends, so of an unknown sequence. But using long-range information from different technologies, you will be able to bridge the gap between the contigs and put them together into larger scaffolds. So the first step is a scaf-10x using 10x data. 10x data is essentially a library preparation method that is followed by classical Illumina sequencing, where you can actually barcode you can barcode individual DNA molecules using gel beads and a microfluidic chamber. So you end up having essentially your final reads that have a specific barcode, and all the reads that share the same barcode, you can be sure or almost sure that they belong to the same original DNA molecule. Then the second technology that is used for scaffolding is bio-nano-optical mapping. So in bio-nano-optical mapping, essentially you take your DNA molecules, you let them flow through these nanoscale arrays, and before loading them, you have been able to stain them to recognize specific motifs along the DNA, and you can then use this information to again to link together contexts. So this is how they look like, if you haven't seen them already. And the last technology that we use for scaffolding is Hi-C. So Hi-C is actually cross-linking the DNA within the cell nucleus. So before, essentially, instead of extracting the DNA, you allow this cross-link to occur within the immobilized DNA within the nucleus, and then you essentially is going to tell you how likely it is that two sequences that are close to each other in the 3D space are also found together in your assembly, and therefore they should go together in the assembly. So this is a summary, as you can see. So this is, let's say, the genomes we're targeting. We generate long reads, so sequencing reads. Out of them, we can generate the context by finding the overlap, generating the consensus out of the overlaps. Then we can use these bio-nano maps to essentially put together more context into a single larger scaffold. And then we can also use this information from Hi-C reads to essentially bridge the gap between larger scaffolds. Here is just a summary on how much scaffolding these technologies allow. So PacBio, as you can see, you have the link distance that is around 10,000 base pair, a bit more. Than 10x allows you to increase a bit the size of your scaffolds. Then you will have, these are all bio-nano technologies. Now we're using this DLE1 enzyme, you can see performs better. And then you have the Hi-C data, which also allows for further scaffolding. And in many cases, this allows you to reach chromosome level scaffolds, which means that both, say, chromosome arms are represented in your final assembly for all your chromosomes. There will be gaps in it, but nevertheless, you will be able to tell this is a chromosome, or it looks like it's a chromosome. So the first step. So this is how a workflow within DNA Nexus looks like. So normally you have your inputs on the left, then you have some parameters that you can configure for that particular step or app or workflow, and then you have the outputs that are generated. So for instance, in this CAFTANX, you will have to provide your assembly, so your primary assembly, and also you will have to provide your row 10x data. So this is like forward reads and reverse reads. Then essentially when it says set inputs means that there is nothing else to provide, and then you can run the clicking here. Okay, and when you click there, it's going to run. It's going to take maybe, I don't know, 1 day or so. And then after that, you will have your outputs, which include the scaffolded context. One important thing I would like to mention here. So in order to make sure that you're actually doing everything in the appropriate way, the most important thing is always to try to calculate the N50 contigs or N50 scaffolds, all these metrics that I mentioned before, at any step of the pipeline, so that you make sure there has been not, like, say, a problem with, I don't know, the input files, or there was something unexpected that happened, because you should expect to see some improvements, let's say, in your statistics. If you don't see any, then you should contact us or review it anyway. So one important thing is like to try to review as much as possible all throughout the steps of the pipeline, anything that you see. Then one thing here that I mentioned is that at any time of the pipeline, your internal files from the Nexus back to the cloud, so back to our GenomeArk bucket. And therefore, you can potentially use this like a sort of intermediate backup. And of course, whenever you do that, everything will become public, so it will be available for anyone to download. I think there is some noise in the background. I don't know if I'm the only one hearing it. Okay, that's good. Sorry. So then as to the scaffolding, the second step, the first one was Scaffold-Next. We'll be using Bionano optical maps, as I mentioned, and as I said, we're now consistently using this DLA1 enzyme. This is the outlook of the Bionano scaffolding pipeline. Instead of providing, of course, the 10x data, you're going to provide the Bionano data. Consider that for all the projects that you're going to run, there should be everything that you need available within DNA Nexus. So there should be a folder for each data set. And then similarly, you're also providing the scaffolds that you want to scaffold further. And then essentially the pipeline has already been set up mostly by Maria and Chai on DNA Nexus. So we'll take care of everything everything that is needed. Of course, there will be a lot of fine details that I'm not going to review here, but for anything that you need, you can just ask me or anyone in the training channel, me or Rang and so forth, for clarification or for any doubt that you have, or and this will happen from time to time, for errors that you encounter while running some of the tools, because of course, depending on the genome that you have, there may be some parameters to adjust. So if you click here on the gear, well, that's just a detail, but that's also for the previous 2-enzyme approach, but now we're consistently using a single enzyme approach, so you don't have to be about that. Again, after this step, please run the assemblies so that you make sure that you see an improvement. And if you don't see any improvement, that's maybe possible, because sometimes that can happen, but we can see what happened and make sure that was actually normal. Fourth, scaffolding. So that's actually the third scaffolding step. So just to remind you, Scaffold-Next, Bionano, and Salsa. Salsa is the one is this software that is actually taking the Hi-C data and using them for scaffolding. Can you still hear me? No.
Good, everyone.
Oh,
okay. I can hear you well.
Okay, thank you. So, so this is scaffold. This is the third scaffolding step. What we actually, the convention use is that the purge dupe step is the first scaffolding step. Anyway, as you can see again here, you're providing your genome assembly, you're providing the raw data. Sometimes in this case, for instance, you have to provide the restriction sequence that was used to generate the library for Salsa, for Hi-C. So you need to provide Salsa with that particular information, but it's something that you should find within the raw data folder. There should be a text file providing that. So for instance, in ARIMA, these are the two options that you have for dovetail and phase. This is the only recognition sequence that is used, so depending on your data set. Again, calculate the assembly stats. Sometimes, in many cases, you will have to, let's say, change the name of the intermediate files that are generated to match what was the conventional names that we're using for intermediate files. That's something that is provided in one of these slides, so you don't have to worry about that. Then we have a polishing and curation step. So after 1, 2, 3 scaffolding steps, we get to this S3 file. So these are the names, the conventional names for our intermediate files that I was talking about just before. And then when we get to the S3, we are actually performing a polishing. So polishing means align all the reads that were generated from PacBio and 10x, let's say the supporting evidence of our assembly, and see how well the raw data support the final assembly. And whenever they don't support it, we're going to change the base that was maybe introduced by mistake in the Falcon assembly step or somewhere else. And also here, importantly, there is one so-called gap filling step. I won't go too much into that, but essentially this arrow polishing step is taking the long reads and essentially is trying to fill in the gaps that were introduced by the scaffolding steps. As I said, scaffolding means take your contigs that by definition have no gaps, no ends, no rounds of ends within them, and trying to fill in these gaps by using the long read information that now by mapping everything back to the assembly may actually being able to fill in these gaps. So there is one arrow polishing step, and then there are two freebase polishing steps which are using the 10x data. 10x, so these are the canonical short reads from Illumina, but with the barcodes. So this improves the quality of the mapping of the reads to the assembly, and therefore it also improves the quality of the final polishing. As you can see, so in this particular step, that's probably worth mentioning it now. So there is, as I said, we're only scaffolding the primary, but the alternate are then combined back to the primary in this particular step to make sure that you end up having both, let's say, all the haplotypes that are present, both maternal and paternal, and are available for the long reads and the short reads to map to, because we are not, I mean, the raw data contains both the maternal specific sequences, and this is done to avoid over polishing some regions of the genome using information from one haplotype to polish the other haplotype. And then after we have completed polishing, this is probably one of the most expensive steps, and also the ones that is taking longer. Then what we do is we essentially separate back the primary haplotype from the alternate, and the primary, of course, is going to be your real, let's say, reference assembly. The alternate is just going to represent what was not part of the reference, let's say your SNPs or your structural variants. And then here is where most people in this call will stop, because then what happens is that you will send your assembly for curation to a highly talented team at Sanger Institute that is going to take care of trying to see and validate every single let's say sequence of your assembly by looking at all the data, map it back to the assembly, and see how and manually actually trying to look into how much what is the level of consistency of your data with the assembly itself. And then by curation, you will end up having this primary and alternate curated assemblies. That is what we are going to submit to NCBI and to all the archives for annotation of the genome, and also for like public reference. So within this step, as I said, there is one step where we are using the raw bio reads. Sorry, one thing that is important here is that we have noted relatively recently that okay, so there is a problem. The mitochondrial DNA, of course, every vertebrate should have also a circular mitochondrial DNA sequence, but the way all algorithms for assembly work, so Falcon and so forth, are actually targeting a long linear sequence that is our chromosomes in the assembly, and so they are not always dealing well with any small circular element like the mito sequence. So therefore what happens here is that you have a problem in trying to assemble it, and what we have developed to overcome this issue is a alternative pipeline that is specifically targeting the mitochondrial sequence and is trying to assemble it. And what happens is that, imagine if you don't have any mito contig, so if you don't have any contig that is actually representing your mito sequence in your assembly, all the reads that actually were originated from the mito will still be there, and in many cases they are very abundant because there is some sort of enrichment for the mito within the library preparation, in many library preparations. So what happens is that you will have all these reads that when you try to map them back here in the hour polishing or in the freebase polishing step, when you try to map all these reads back to the assembly, they will essentially map and somewhere else in many cases, because they will not have anything that is really homologous to map to. Okay, so what we try to do here is that we assemble the mito separately and we add it before we polish, so that we avoid having these over polishing of unintended regions of the genome. So I'm working, myself on this. So whenever you reach this step, just ask me or look up in the GenomeArk if the mito sequence is already available, and therefore you can add it to your assembly. That's something we can, review throughout the process. So after having added the mito, you can start the polishing. And what happens here is that you have your, first of all, you have this workflow from DNA Nexus, which is called Scaffold 5 PacBio BAM sequencing workflow. And as you can see, it's essentially taking all the sub-read BAM files, which are the raw data from PacBio, and is mapping them back to the reference genome and trying to use these mapped reads to improve the quality of the individual base calling of your genome assembly. Again, calculate the statistics, because as I said, there is this gap filling function within this particular step, which may actually improve the quality, the contiguity of your assembly at this particular stage. Then, as I said, we polish also with 10x data, so you can take all your 10x data and map them back again. We use this Long Ranger, which is the proprietary software from 10x Genomics, to align the reads. And then we use this variant caller to check the consistency of the alignment with the individual base calling within the genome assembly. We actually run 2 times this step here. As you can see, there is this 2x. We ran it 2 times, and we have, in order to reach a final consensus sequence that, as I said in the very beginning, we are targeting QV40. So the individual base calling should be QV40, that is less than 1 error every 10,000 base pairs. And that's why we run 2 steps of it, because we have seen that this usually reaches QV40. Steps is also taking care of actually trying to calculate the number of errors in the assembly at the very end of it. I will not go into that, but if you're interested in how to do that, we can review it later. So yeah, so this is again the workflow for this particular step of polishing. So again, you provide your assembly at this stage, and you also provide the original raw data from 10x for alignment, and then it's going to take care of everything else. So yeah, well, you have to provide some few, like I don't know, the genome ID and that sort of things, but mostly you don't have to do much. Okay. And so after this step, let me just go back to the pipeline. After this step, as I mentioned, we're almost done. So we get the primary and we just need to send it out for curation. However, it's it would be nice if we can, let's say, review somehow ourselves the final assembly before we send it out to the Sanger for curation, to make sure that everything looks fine. So there has been no like misassembly. Of course, there will be errors in the assembly, but nothing that was like an error in running the pipeline. First of all, of course, we are going to. You can go. Most of the people, I think, are already have access to this table, but if you don't, I will grant you access. So within this particular table, we're tracking, as I said, all the steps that the assembly is going through, and we are also tracking the some of the metrics that the assembly I mean, that all these steps are achieving throughout the assembly process, which means, like, for instance, this is just the first one is like the contigs straight the assembler, so out of Falcon. Then what happens when you run purge tubes, when you run Scafton-X, when you run Bionano scaffolding, Salsa for Hi-C, you run the polishing steps. And so you can track down everything that's going on, and you can also, I mean, infer if there is any issue going on. For instance, if you see that your final QV is very low, there may be some problem with the raw data that was used or something of the like. So I will, in the video, also show you how to update this table, but basically that's very important that we keep track of what's going on at every single step. I'm sorry, I will close this one too. Okay. So, so this is an example where you see that essentially in C1 you had a N50 of 25 megs. C2 is actually the name of the alternate haplotypes. So as you can see, that's much smaller in most cases, and by definition it has to be somewhat smaller. Then you expect some improvements with Scafton-X, some further improvement, usually quite good improvement with Bionano, and also sometimes, but not necessarily, you see an improvement in terms of N50 when you run Scafton-X. Here it is important to state that even if you don't see an improvement, that doesn't mean that there's been a problem, because first of all, the N50 is measuring something, but is not necessarily measuring everything that has happened. And in this particular case, I can predict that maybe there has been some scaffold that was joined together, but this is not recorded by the N50. So that's something to double check, but doesn't mean that there is an error there. But for instance, in this other case, you see that you go from 42 megs to 222 megs. So there's been a sharp improvement in this case. So there are some more complicated checks that one could actually run at the very end of the assembly process before sending it for curation. So essentially to, let's say, find out if there was a problem before it takes maybe a while for the people at the Sanger team to find out themselves. So one thing one can do is actually try to see how the Hi-C data looks like when you map it back. I'm sorry, I don't have an image for that, but when you map it back to your final assembly, and if there is consistency between the Hi-C data, so this short read data that is providing you long range information with the assembly, then you could also, that's something actually run also at the very beginning of your assembly and see if there is like an improvement between the beginning of the assembly and the end of the assembly, especially after polishing. And it's the Basco scores. So Basco is available on the NNXO, so it's just something you can configure and run in 5 minutes. It's going to maybe take 1 day or even more, but it's very easily to start, to run. And it's going to tell you how many genes that we expect to see in your assembly are actually there. It's based on a conserved set of genes that was by who has developed Basco, and by looking at how many genes that you expect to see in your assembly because they are, let's say, common and shared to all vertebrates or to all fishes, and how many of them you see, then it's a proxy of how complete your assembly is. And normally you should see that there is over 90% completeness, but it can also go up to 97. It also depends on, several factors. But what also is very important there is that you see an improvement between the initial assembly and the final assembly, because this will be also a function of how the base calling quality is. So the more accurate the individual base calling is, the more Basco genes will be identified and therefore will be assigned to your assembly. Then of course calculate all the assembly statistics, calculate QV. Remember, we are targeting QV above 40. So if you don't get QV above 40 at the very last step, please tell us, because there might be something going on. And then, well, yeah, that's pretty much it. Then there are some like more sophisticated analysis that one could actually carry out using k-mers. Probably you're not going to do that, but sometimes it could be, especially if we notice that there might be an issue, we could actually try to see if this issue somehow is recapitulated in the k-mer analysis. You may also want to, let's say, plot your assemblies. This is not a very good example, I think, but you can actually see how many, let's say, how syntonic your assembly is compared to other assemblies from the same or closely related species. As I said, we can run Basco. Many of these things actually will be also done at Sanger directly, but as I said, instead of wasting like time trying to like ping pong between the Sanger and us, we can also try to run some of these ourselves. One thing, how many? Yeah, so I think we can maybe have another, I think maybe 10 minutes or even less than that. And then when, I mean, all the questions that you may have or anything you want to discuss or anything you want to correct that I may have stated wrong, we can actually look at that. So this is about exporting to GenoMark, which means moving the files, intermediates or final files from the DNA Nexus, where you perform all the compute steps, to the GenoMark. Okay, so that's pushing everything to GenoMark. It's straightforward again. So in this slide, I'm not going to go through it, but also the slides will be available for you to review at any time. But the point is that before we actually upload and make it publicly available, we just like make sure that the folder structure is correct, that file names are as they are expected to be. And then there is a very simple app. So you see here, install apps, DNA Nexus to VGPS3 exporter. Just that is actually taking care of just going through your project on DNA Nexus and move all the files from DNA Nexus to the GenoMark. And when you move the files, they will not disappear from DNA Nexus, but there will be a symlink, so a sort of empty copy of the file that you may, in many cases, still be able to use and visualize on DNA Nexus. So, I mean, one of the first checks is just make sure that the directory looks as you expect it to be. If there are like files to rename them, rename the intermediates as well as the last files, but that's something, again, we will, when we reach that step, we're going to review it. Also, if you want, you can, I mean, some people will like run the entire pipeline and then export everything at the end of it. Some others will, at some point during the pipeline, run the exporter directly. This is helpful from our side because this allows you to actually reduce the storage that is required from us on DNA Nexus, which is also saving money a bit. So that's up to you. I mean, you can feel free, depending also how long it takes to you to run the whole pipeline. Consider that for small genomes as these, maybe in 2 weeks, even if you're not like working constantly on it, you can actually make it. So yeah, that's pretty much it. This is how this app to export from the DNA Nexus to the AWS bucket is actually working. As you can see, you only have to provide the species ID where you want to export things to. Yeah, so this is something I already introduced. So just a few checks before you send out for curation. Are the files really available for download? Is the QV in the expected range? Is the genome? So can we make sure that sometimes there has been maybe species misidentification in the first place? So at some point of the pipeline, we can actually make sure that using essentially, I don't know, mito data or existing information from the same species, that we are actually looking at the species for our assembly, or I don't know, sometimes one can mix up the sequencing data before uploading them. So that's also a possibility, just to make sure that they are how they look like, they look like as expected. And as I said, you can generate this very nice IC plot. That's also something we can review together. It's not particularly complicated if you have some compute. And then this is the final step where you actually send out for creation. So you just email. They have a very nice tracking system at Sanger. So you just email them with a specific email stating some of the summary information from your assembly, and you're done. So that's pretty much it. It was 1 hour, I think. So I will now, I think you can probably unmute everyone, and if there are questions or I don't know, points you want to be discussed, we can do that. Thank you.
So, I have a comment regarding your last slide for the Q&A. Can you go back one slide?
One.
So
here.
No. Regarding the final check before sending for
this one.
Yeah, this one.
Okay.
So Kirsten from the curation team to generate Hi-C maps for all the assemblies before sending for curation. And there is another app on DNA Nexus, another workflow that just runs the Hi-C alignments. So we will follow up with that.
Oh, okay.
To that stage.
So this means that you will not have to generate the alignment locally yourselves, but you can just use DNA Nexus as well for generating the Hi-C alignments, which is something that the curators will use for evaluating the quality of the final assembly.
Should add is actually two things. The first thing is once you send the email that, if you're on the next slide, the email to Grid Jira, make sure that you do get a response back from the system. If we don't get a response back from the system, it means your email hasn't gone through. That's number one. And number two is that when the first person you will probably interact with via the system is my colleague, James Torrance. And one of the things that he does when he receives your request is he runs it through his contamination pipeline to see if there's any contaminants from different samples or remaining adapters. Right now, it's pretty good, but you never know. This is a step that needs to be done, and because this step is done, there are times when the assembly coordinates may change, and hence why some things like Hi-C or remapping of the original data has to be rerun on our end in order to fit the analysis that we need.
Got you. So does anyone ask questions on the pipeline or how to get started? I don't know.
A few questions.
Sure.
Can
you tell your name?
Camila, here from Berlin.
Oh yeah, sorry, because I can't see you. Okay.
Are all the steps from the pipeline now implemented in DNA Nexus and working?
Yes. So now the pipeline, so one thing is that it took a while to actually migrate the pipeline from, let's say, the local version running on this LARM cluster to DNA Nexus. But now I think the transition is essentially complete. There may be like some supplementary analysis that are still to be, let's say, migrated, but essentially all these steps that I've been going through are now working. I can testify myself because I've been running it. So you should not need to run anything locally.
Okay. Do you know if there are different labs who are running things locally?
Well, we are running it locally here at the Rockefeller as well. So we are also doing it locally. So if your question is is there any anyone to refer to if you're running it, I mean, part of it or entirely locally for some projects? You can ask me. I think, of course, you can ask Arang, who has developed the pipeline herself. So that's for sure. But yeah, there are a few people that are running, and also at Sanger Institute, by the way. I mean, all those that are run on DNA Nexus, they are from projects that are not directly from Sanger because they are running it locally themselves.
Okay.
So all the projects that gets funded for the computing is gonna go through the DNA Nexus pipeline. And for the rest, it requires a personal commitment and a cluster for sure. So in those cases, you can use the local version of the pipeline. It's essentially running the same steps.
Does it matter if maybe versions of some part of the software might be different between DNA Nexus and what people are running locally? How much of an effort should everyone make to try to use the exact same versions?
It's better to run the latest software whenever possible.
Mm-hmm.
The DNA Nexus pipeline is almost running the latest versions for all of the steps. There are a few steps that Chai is planning to update to the latest version. So I would just say run whatever the latest version that's available.
Okay.
And whenever, of course, I mean, should you have any doubt on that, I also sometimes have doubts on the versioning of some tools. So just feel free to ask.
Okay, that's great.
Is there some kind of guidelines or GitHub where you can access to download the latest version for the local computing?
I'm sorry, I'm not sure I got your question correctly.
My question is if any GitHub or I don't know, repository where I could go and look for the latest version of every software run locally?
Yes, so the GitHub page, I'm not sure if I can quickly find, yeah. So this is the pipeline, okay, with the latest version for all the steps that you may run locally, okay? So essentially, let's say if your Slarm works exactly as the one that was used to develop the pipeline, you can run every step just by downloading the GitHub and running each individual step one after the other, and you should have no issue there. Maybe there is like something you have to look into if it doesn't work. There is also that's not for the local version, but for DNA Nexus, I think we are going to update a sort of tutorial which goes step by step throughout the pipeline and allows you to, know whatever you need to do at each individual step.
Yeah, I was asking about that. Like for DNA Nexus.
Okay. Any other comments or questions?
I need to update that README file actually.
Yeah, I know.
Yeah.
So the current I think it's using 1.5 version probably, and so there is and also it's probably missing one of the last steps of the pipeline. But I think we're gonna update it. Also, it's very well written, so it's quite clear what you have to do at every step. So as soon as it's updated, we'll send an email. And
So currently it's structured in the tools name, like each folder under the VGP assembly pipeline has a folder name of the tool that you're running. And under that directory, you can find a file that's a submitter repr. It starts with _submit. It's developed on a Slurm-based cluster environment. You can convert that to your own, match that to your own cluster environment if it has a job scheduler. then it will, inside the submitter script, there is the script that is actually running. So yeah, if you need to adjust the code, you only need to copy the submitter script and edit that to your own version at the moment.
Right. So just to get started, as I told in the beginning, I'm sure there is like this list of people at the very least that will, I mean, has either already started or is going to start very soon. I am aware that there are a bunch of people. Well, one is from Berlin, but also another person that is willing to start trying to assemble one genome. And I think I have 2 genomes that are available for that. If there is anyone online, or if there is anyone when, let's say, you listen to these recordings that is willing to assemble one genome or trying to assemble one, just send me an email so we can see if there is anything available. So to get started, you can, well, if you don't have any account on DNAnexus, you should first just Google dnanexus.com, get there, create your own personal account, then let me know so that we can allow you to run analysis on your account for us, for the VGP community. Then you can maybe also, if you want, you can start familiarize a bit with like the project layouts, and then maybe when you want to get started, just ping me so that I know, and we can maybe have a look at that. The first step in most cases is you start from Falcon, so Falcon to generate the contigs, unless they have been already generated locally, like in the case of fishes, and then also in the case of trios, because that is not implemented on the DNAnexus as well, but maybe it was going to be implemented at some point. And then one thing is like calculate the assembly statistics for the contigs, or also you can run BASCO analysis. As I said, that's available in DNAnexus, so you can search for BASCO within the VGP tools folder that I mentioned at some point, where we essentially have all our applets, and here it is. So if you search within the projects on DNAnexus, you will find these VGP tools. Within VGP tools, you have all the applets that have been developed specifically for the VGP, as well as all the workflows that are used. And there is also one applet here that is running BASCO, so you can test that too. The other thing is that you can estimate the genome size. As I said, jellyfish is not necessarily working well as predicted. I mean, it is a good estimate of the genome size in some cases. So if you want to run that, please let us know, because we are going to, I think at some point, I mean very soon, also release for DNAnexus another tool that should be more effective, and it's actually more effective in estimating the genome size. So, and as I said, that's it's something that you essentially, when you calculate the statistics throughout the scaffolding, it's something that is normally required by the software. And as I said, when you run Falcon, it's also very important to provide a decent estimate. And when the repetitive content of your genome is very high, jellyfish is very poor at that. So you may have like a genome size estimate that is 1/5 of your genome assembly size. And that will be a problem if you run it with that estimate.
Another question about the chromosome numbers, because you said you should have at least 90% of the data should be assigned to chromosomes.
Right.
How can one know that? How can you know how many chromosomes there are? Which ones would be the chromosomes?
Yeah, so it's not something that we take care of when we run the pipeline. So it's something that is for the curation team. Mm-hmm. But maybe Arango, you want to comment on that a bit?
Yeah, so the chromosome assignment happens during curation. So when the curators pick up the structural abnormalities, given all the row sequencing data signals. And they look into the closest species that's available. For example, if there was already a Sanger Illumina-based assembly available that has been chromosome assigned, those chromosome numbers will get carried on. Or we could take the closest species genome, which when it is available, or using linkage maps and such on. If there are no such reference available, then I think the largest scaffold is named 1 and after like that as scaffolds. Yeah, I'm not sure if that was chromosome assigned. I think, yeah. And
So-
during that, trying to find the sex chromosomes as well, so we don't. I see Joe there.
Hi, just to throw in, so we try where possible to find previous karyotype information, which has proved quite successful for a number of the species that have come through so far. Where we can't find the actual species, we look at family information, if there's anything available there. The Hi-C itself is very good at identifying individual chromosome constructs, which is usually a pretty good guide of what the expected number should be for the haploid. Obviously, that's not always possible, because, or it's not always, karyotype information isn't always very accurate. I think with the sloth, the karyotype that's available is wildly different to what we're seeing from the actual assembly.
So it's still, let's say, under development, but that's the target. I mean, and I think in most cases you actually achieve that.
Yeah.
Yeah.
Something for the people who are assembling to worry about, right? Okay.
Yeah. When you get the stats, like for each steps, I saw there was a confusion.
Okay.
A trainee saw that, oh, my scaffold N50 reduced after Bionano scaffolding, and then he thought, oh, maybe my assembly went wrong, and he skipped that step. But that is actually happening sometimes. Bionano and Salsa, they are doing breaking first and then doing the assembly when they see confident evidence of misassemblies. Don't be surprised and try to get rid of one step. Most of the time, unless you have very low coverage, most of the time the assembly scaffolding steps are doing its job. So when you see those kinds of things happen and are not sure whether you can rely on that outcome, just ask on our Slack channel to be sure.
Right. So here, for instance, you can see that from S1, which is Scaftenex output, to S2, which is the Bionano scaffolding output, all cases are actually increasing. But since Bionano can actually cut the DNA, it could also decrease. Yeah, just ask.
Is there a way to estimate the quality of the Bionano data or Hi-C data so that, I don't know, if there is a huge improvement, one would maybe know that, oh, this is because of low quality of some of the types?
Yes. So, well, for Bionano, we don't have a in-house developed tool, but the point here is that you will receive Bionano CMAPs, which is essentially the consensus out of all the individual Bionano molecules that was generated previously and uploaded directly to the project. So my point is that someone should have already checked that, so that the Bionano is that data set is fine. And the same goes for all data types. So the assumption here is that the data you're dealing with is good, and therefore the error should lay elsewhere. That's not necessarily the case. So if you like, the methods are not changing, we can review that. But on average, we're not uploading data that has not like reached past the quality controls.
Yeah. Well, sometimes we have seen when the data was generated a long time ago and came into the pipeline, sometimes the Hi-C data maps were in a good quality. The coverage, the depth of coverage and everything looked fine, but when we aligned the data back to the assembly, you could see that from the Hi-C map, everywhere was just giving a signal, or you get barely any signals from anywhere, meaning that you sequence perhaps something else. So yeah, for Hi-C, you can easily visually identify whether your Hi-C data is clean and nice. I just wanted to add that. Yeah, I don't know if we have to run mesh screen on these newly assigned genomes, Julio. Perhaps we need to Martin if they were screened first.
Yeah, that's a good point. So whenever we receive something from like that was already generated, so we have not generated ourselves, it's actually a good thing to make sure that there is no like abnormality there. So this mesh screen that Orang is talking about is actually essentially trying to look how the raw data that was generated, let's say PacBio raw data or 10x raw data, so sequencing data, how is consistent with each other. So how, let's say, one SMRT cell or one Illumina flow cell is similar to the other that was run on the same sample, assuming that you should see the same results from two different flow cells that were using the same input DNA. So yes, that's something we can actually do here. So it's okay. It's a good check, because the problem, of course, if you're using the wrong raw input data, and I haven't mentioned that clearly, but all these steps that we run on DNA Nexus have a cost associated to them, which actually you normally can monitor. So when you see a job running, you will see how much it costs. So the problem, of course, is that if we realize only at the very end of the pipeline that there was some problem with the raw data, of course, it's hard. I mean, it's going to be expensive to rerun everything afterwards. So yeah, it's a good check. We can actually look into that.
Yeah.
So I think, I mean, unless someone else has other questions, we are almost on top of the hour, so we could.
Yeah. Hi, Julio. This is Rohit from Berkeley. Can you hear me? Yeah. So I had a question. So we were trying to sequence one rockfish, which was highly heterozygous, and Falcon has not been the best solution for us. And we don't have trios, and this is wild rockfish, and we just sampled it from the ocean. So we were wondering if you had any other alternatives for the genome assembly apart from Falcon or Canute Trio. Mm-hmm.
Yeah, so the problem with highly heterozygous genomes is that you get something assembled that's much larger than your expected genome size. And I think you will see the same regardless you assemble it with any assembler, either Falcon, Canoe, or other or whatever. That's because when the regions are too much highly divergent, the assembly picks it up as a different part of the genome and just assembles it and assigns its own contig to them. So for those cases, I think purge dupes will be a bit more effective. So if you already have a Falcon assembly, you could try run purge dupes on the primary set and see if that matches your expected genome size. Or you could, if you think your genome is too highly repetitive and the contig M50 were a bit disappointing from Falcon, you could try Canoe or other assemblies. And of course, those are not designed to unzip your haplotypes. So you have to do the unzipping yourself. So that's where purge dupes can get in and help you.
Okay.
Yeah.
So we have been testing that DBG2, sorry, not DBG, WT-DBG2.
Yeah.
Yeah, so when we are playing around with parameters, we have identified that when you use Canoe corrected and then throw it into WT-DBG, it's much better.
Mm-hmm.
But I was not sure at the VGP if you recommend this or if this is, yeah, not.
Yeah, I think that's a fair way to do. Yeah, I wouldn't stop you from using that assembly. The only version with WTDBG we had was that we saw a bit more collapse going on.
Yeah, we tested this too, and when we were changing the parameters for the Kimmer collapsing and the adjacency, it looked okay.
Yeah.
In terms of duplications or over collapsing, we were able to adjust the parameters and it was performing better.
Mm-hmm.
Okay, all right, thank you.
Mm-hmm.
Thank you so much for the explanation.
See you.
And in case we have we need some further discussion, we'll just going to have another Skype. Okay. Bye bye. Have a nice day and a nice weekend.
Thanks, Julio.
Bye.
Bye, everyone.
Bye.